Today we are going to learn about the neuroanatomy, the introduction of nervous system. The neuroanatomy, it is a study of nervous system. The nervous system is the most complex, widely investigated and least understood system in the body. It along with the endocrine system regulates the functions of the all other systems of the body. Hence, nervous system is called as master system of our body. In this session, we are going to learn about the introduction, divisions of the nervous system, cellular architecture, neurons, structure of the typical neuron, classification, nerve fiber, and neuroglial cells. The nervous system is the chief controlling and coordinating system of our body. It adjusts the body to surroundings and regulate all bodily activity, both voluntary and involuntary. The average weight of the adult brain is in the air it is 1500 grams in the adult male. In adult female it is 1300 grams and it consists of about 200 billions of neurons in adult brain. Next, the divisions of nervous system. The nervous system which is divided into two. One is anatomical and functional. The anatomical division which is divides into central nervous system which comprises of brain and spinal cord. The CNS is seat of learning, memory, intelligence and emotions. Then second one it is called as the peripheral nervous system or PNS which includes 12 pairs of cranial nerves and 31 pairs of spinal nerves. And these provide afferent impulses to the central nervous system and carrying efferent impulses to muscles, glands and blood vessels. Next, the functional classification. Nervous system functionally classified into two. The afferent component provide sensory informations to the CNS and efferent component carry motor informations to the muscles, gland, blood vessels and heart via somatic nervous system for the control of skeletal muscles, autonomic nervous system for the control of heart, smooth muscles of the organs, glands and blood vessels. Next, the cellular architecture. The nervous tissue is made up of nerve cells or neurons and neuroglial cells or neuroglia forming the supporting or the connective tissue of central nervous system. The neurons are specialized cells that constitute the functional unit of the nervous system. Next, classifications of neuron. The neurons are classified according to polarity and according to the length of axons. According to the polarity of neurons, which is classified into unipolar neuron, bipolar neuron, pseudounipolar neuron, and multipolar neuron. The unipolar neuron has a single cell body and a single cytoplasmic extension, which commonly present in the mesencephalic nucleus of trigeminal nerve and also occurs during the fetal life. These cells are more common in lower vertebrates. The major examples are dorsal root ganglions, sensory ganglia of cranial nerves. The bipolar neurons, it have a single cell body at the center and two cytoplasmic extensions. One is considered as axon, another one is dendrite. This is commonly seen in retina, sensory ganglia of cochlear and vestibular nerve. The pseudounipolar neuron has a single cell body and a single cytoplasmic extension. Immediately after that, that forms a T junction and which gives two right and left branches. The one is called as the axon, another one is called as dendrites. So it is called as pseudounipolar neuron, which is located in the dorsal root ganglia of spinal nerves and sensory ganglia of cranial nerves. The multipolar neuron has a single cell body and numerous cytoplasmic extensions. The shortest cytoplasmic extensions are called as dendrites, and it have a long cytoplasmic extension. It is called as axon, which is commonly located in the motor neurons of anterior gray column of the spinal cord and autonomic ganglia. According to the length of the neuron, they are classified into Golgi type 1 and Golgi type 2. The Golgi type 1. This neuron has long axons and numerous short dendrites. These are seen in pyramidal cells of cerebral cortex, Purkinje cells of cerebellum, and anterior horn of cell of spinal cord. The Golgi type 2. These are the neurons with small axon and established synapse with neighboring neurons. These are also seen in cerebral cortex and cerebellar cortex. The structure of a typical neuron. It has a cell body and neurites. The cell body, which is otherwise called as soma. In the cell body of a neuron contain, and centrally there we can see a nucleus and endoplasmic reticulum, mitochondria, Golgi apparatus, and axon hillock. The cytoplasm gives numerous small extensions which is called as dendrites. The dendrites ends in a minute branches which is called as dendritic branches, which is joins with another end of an axon to form synapse, and it have a longest cytoplasmic extension which is called as axon. Then at the end of the axon, which is divides into minute branches and which forms a synaptic terminal and which is joins with another dendrites. Next we are going to learn about nerve fiber. The nerve fibers are formed by axons or some dendrites which resembles axons in structure, which constitute what are commonly called as nerve fibers. The bundles of nerve fibers found in the central nervous system, which is called as tracts, while bundles of fibers found in the peripheral nervous system, which is called as peripheral nerves. The basic structure of a peripheral nerve fiber. Each nerve fiber has a central core is formed by the axon. This core is called as axis cylinder. The plasma membrane which surrounds the axis cylinder, which is called as axolemma. The axis cylinder is surrounded by a myelin sheath. This sheath is in the form of short segments that are separated at the short intervals called as nodes of Ranvier. The part of the nerve fiber between two consecutive nodes is called as internode. Each segment of myelin sheath which is formed by one Schwann cells. Outside the myelin sheath, there is a thin layer of Schwann cells cytoplasm and an external lamina. This layer of cytoplasm and external lamina is called as neurolemma. Each nerve fiber is surrounded by endoneurium. This is a layer of connective tissue. The endoneurium holds adjoining nerve fibers together and fasciculate their aggregation to form bundles or fasciculi. Each fasciculi which is surrounded by the perineurium, that is at the thicker layer of connective tissue. The perineurium is made up of layer of flattened cells supported by layers of collagen fibers. The perineurium probably controls diffusion of substances in and out of axons. A very thin nerve may consist of single fasciculus, but usually a nerve is made up of several fasciculi. The fasciculi are held together by epineurium. This is a faintly dense layer of connective tissue that surrounds the entire nerve. Classifications of peripheral nerve by two, according to the function and according to the area of innervation. According to the function, which is classified into efferent and afferent. Some nerve fibers carrying impulses from spinal cord or brain to peripheral structures like muscles, glands, and they are called as efferent or motor fibers. Other fibers carrying impulses from peripheral organ to brain or spinal cord. These are called as afferent fibers or sensory fibers. According to innervation, they are classified into somatic sensory fibers, somatic motor fibers, visceral sensory fibers, and visceral motor fibers. Somatic sensory fibers, they convey impulses from skin, bones, muscles, and joints to the central nervous system. The somatic motor fibers, they carrying impulses from central nervous system to the skeletal muscles. The visceral sensory fibers, they convey impulses from visceral organs and blood vessels to the central nervous system. Visceral motor fibers, they carrying impulses from central nervous system to cardiac muscles, glands, smooth muscles within the viscera. Myelinated and non-myelinated nerve fibers. A myelinated nerve fiber is one that is surrounded by myelin sheath. The myelin sheath is formed by oligodendrocytes in central nervous system and Schwann cells in peripheral nervous system. Small diameter axon, for example, those of the ANS and small pain fibers are simply enveloped by the cytoplasm of Schwann cells. These nerve fibers are said to be non-myelinated. An axon lying near a Schwann cell that invaginates into the cytoplasm of Schwann cells. In this process, the axon come to be suspended by a fold of cell membrane of Schwann cells. This fold is called as mesaxon. In some situations, the mesaxon become greatly elongated and come to be spirally wound around the axons, which is thus surrounded by several layers of cell membrane. Lipids are dependent between adjacent layers of membrane. These layers of mesaxon along with the lipids sphingomyelin form myelin sheath. Outside the myelin sheath, a thin layer of Schwann cell cytoplasm and an external lamina persists to form an additional sheath, which is called as neurolemma. An axon is related to a large number of Schwann cells over its length. Each Schwann cell is provided the myelin sheath for a short segment of axon. At the junction of any two such segments, there is a short gap in the myelin sheath. These gaps are nodes of Ranvier. The part of nerve fiber between two such nodes is called as internode. Non-myelinated nerve fibers are some axons which are devoid of myelin sheath. The non-myelinated fibers are also surrounded by Schwann cells. These unmyelinated axons invaginate into the cytoplasm of Schwann cells, but the mesaxon does not spiral around them. Another difference is that several such axons may invaginate into the cytoplasm of a single Schwann cells. In the TS of nerve fiber, at the center we can see an axon which is surrounded by several spiral layers of mesaxon. At the center we can see the internal mesaxon and peripherally the external mesaxon. Most outside of TS of nerve fiber, nucleus of Schwann cell present. In LS of nerve fiber, when the myelin sheath is examined with the high magnification of electron microscope, it shows alternative thick and thin lines. The thick lines which represent fused cytoplasmic surface of two adjacent layers of plasma membrane, where the thin lines represent the fused external surface of two adjacent membranes. And it's most peripherally the cytoplasm of Schwann cell present. At the center, axon, axonema, and some blood vessels present. In transverse section of optic nerve, we have the ophthalmic artery and ophthalmic vein at the center, and myelinated nerve fibers which is arranged as fasciculi that seen as numerous compartment, and peripherally which is the whole fasciculi which is covered by perineurium. The perineurium is surrounded by epineurium. Next we are going to learn about the neuroglial cells. The central nervous system consists of neurons and glial cells. Neurons constitute about half the volume of central nervous system and glial cells makes the rest. The glial cells provide support and protection for neurons. They are thus known as supporting cells of nervous system. The glial cells does not carry any impulses or action potential. They do not have any important functions. In fact, without glia, the neurons would not work properly. Main functions of glial cells: to support neurons and hold them in place, to supply nutrient and oxygen to the neurons, and insulate one neuron from others, and destroy and remove the cases of dead neurons. Classifications of glial cells. They are majorly classified into two groups: the central neuroglial cells and peripheral neuroglial cells. The central neuroglial cells are astrocytes, oligodendrocytes, microglial cells, and ependymal cells. The peripheral neuroglial cells are Schwann cells, satellite cells, or capsular cells. The major central neuroglia are astrocytes, oligodendrocyte, and microglial cells. First, we can discuss about astrocytes. The astrocytes are largest and most numerous and form the main supporting tissue of the nervous system. They are star-shaped as the name implies and possess many fine dendrite-like processes. At the end of the processes, there are small swellings called as food processes, and it forms 30 to 50% of total brain volume. Types of astrocytes. Protoplasmic astrocytes are found in gray matter. Their processes are thicker and more branched than the fibrous astrocytes. The fibrous astrocytes are found mainly in the white matter. Their processes are long, slender, smooth, and less branched. Further, they contain more filaments in their cytoplasm and compared to the process of protoplasmic astrocyte. Functions of the astrocytes are maintain blood-brain barrier, regulate ion, nutrient, and dissolving gas concentration, and form scar tissue after injury. Oligodendrocytes are smaller than astrocytes and, as the name implies, have fewer processes. They are found in cluster around the neurons of gray matter and adjacent to and along the length of myelinated nerve fibers in the white matter. Function of oligodendrocyte is form myelin around CNS axon. The microglial cells are smallest of glial cells and are capable of migrating through the surrounding neural tissues. Microglia do not develop in the neural tissue. They are derived from phagocytic white blood cells in the fetal monocytes that migrate from the blood into the nervous system before birth. Functions of microglial cells are remove cellular debris and pathogens in the central nervous system by phagocytosis. The ependymal cells lines the ventricles of brain and central canal of spinal cord. The ependymal cells are three types. ependymocytes or epithelial cells, tanycytes, and choroid plexus epithelial cells. The epithelial cells or the ependymal cells are cuboidal or columnar in shape with tuft of cilia on their luminal surface and constitute the majority of the ependymal cells. The specialized ependymal cells in the choroid plexus or the choroid epithelial cells secrete cerebrospinal fluid. The cilia of the ependymal cells assist moving of cerebrospinal fluid through the cavities of brain. And its major function is lining the central canal of spinal cord and ventricles of brain and assist production, circulation and monitoring of CSF. Neuroglial cells in the peripheral nervous system. They are satellite cells and Schwann cells. The satellite cells or amphicytes surrounds the nerve cell bodies in peripheral ganglia and provide support and nutrition to them. The Schwann cells or neuroleumocytes form myelin sheath around axons in the peripheral nervous system. It is important to note that Schwann cells forms neurolemma around all axons in peripheral nervous system, whether they are unmyelinated or myelinated. This is all about neuroglial cells. Thank you.